But let's just get right into it. With our first speaker, we have Dr. Matt Siderhurst. He's a research biologist with USDA ARS at the Daniel K. Inouye United States Pacific Basin Agricultural Research Center in Hilo. That is a mouthful. PBARC. He works at PBARC. His primary research focus is understanding insect communication and movement to improve IPM strategies for invasive species and agricultural pests. He has a background of combining entomology and organic chemistry, and the current objective of his work is the development and use of harmonic radar to track individual insects. The smallest insect being mosquitoes, but to develop novel surveillance and control measures for these tropical insect pests. So take it away, Matt.
Okay, thank you, Jade. And hello, everyone. Let me just check to make sure you're seeing my screen, not in presenter mode. Yay, wonderful. Yeah. So yes, I'm Matthew Siderhurst. I promise that I will not say anything about chemistry today, so I don't want to scare people off for that. That is another hat that I wear. But I want to talk about some work that, notice my name is on the front of this, but this is actually one that was done by a bunch of us. And really this issue where oftentimes when we have invasive species, they come from places where we know something about them. So we get something from China and we're able to go and talk to our Chinese colleagues and say, oh, what do you know about this pest? But the Queensland longhorn beetle is really something different. So when it came out of Queensland and we looked at the literature, it's not really mentioned. We don't know very much about this insect at all. So therefore my title, when you get a new invasive you know nothing about. So really our approach with this study that I'm going to be talking about today is when you get something that's new, how do you figure out just kind of baseline biology, and with the idea of something that we hope then will be helping us with doing control. So I will be talking about kind of 4 different areas. One of them is not really my specialty, so if you could figure out which one that is, well, actually, it's the first one. If you ask me questions, I will probably have to refer that to one of my colleagues. But I'm going to talk about phylogenetic analysis. So this is about the genetics. Scotophase activity is about what they do in the dark. So that's a fancy term for that. Field tracking, which we largely did with harmonic radar, and then some citizen science mapping. So some of you in the audience today may have actually reported data for us, and we're very happy for having that. So the first thing, phylogenetic analysis. So this is work that was done largely by my colleague Shanna here at PBARC. And what she was doing was taking both the beetles themselves and then actually in the end the frass, and looking and see if we can use CO1 mapping and figure out the phylogenetic relationships. So this is other beetles, how are they related? And really the end point for this, the use for control, is that we want to use this for diagnostics. So it's fairly easy when you look at the beetles to say, hey, yeah, we know that's a Queensland longhorn beetle, but it's a lot harder, the adults that is. But when we look at the larvae, it can be a lot harder to tell, what species those are from. And so that's where we think that this is helpful from the diagnostic side, that we can get in there and say, yes, we know that this is Queensland longhorn beetle, or it's potentially another cerambycid. One of the really exciting things out of this too was that they were able to just do this off frass. So frass is beetle poop, right? So if you find beetle poop, potentially we can test that and show whether it's a Queensland longhorn beetle or not. One of the things so this is research that they're actually continuing then. And with that, they've done full genome sequencing for the Queensland longhorn beetle. And it's really interesting when you run that through a number of analyses. We see, hey, that they had a big bottleneck about 18 generations ago, and that fits very nicely with when we know that they came to the islands. So phylogenetic analysis, the first piece that we were doing. The second piece that I want to talk about then is things that we have done looking at their nighttime activity. So I'll kind of draw your attention, hopefully you can see my cursor on the screen, to this setup in C. So we had a large glass arena, and we would put cameras in here. We put a couple of plants in there. We would put some of the beetles in the middle. And then we had an infrared light, which was illuminating it. So in D down here, you can see what some of the views look like. We can actually see a little beetle running around. And so what we were able to do then is watch these videos. And the big thing that came out of this is that one, they are active at night and they don't seem to be active outside of that. So you can see in A where we turn the lights off and we have activity from the beetles, and then lights on and we lose the activity. But also the thing that I think was really interesting for me is that the vast majority of the time they spend resting, or we couldn't see the beetle, so it was hiding, or they're just moving around a little bit. It's a very small portion of the time that they're actually out there being pretty active. So the second one of our studies. So let me move on then to the thing that I'll probably take the longest to talk about, which is the field tracking that we're doing. So this is with harmonic radar. So harmonic radar works by, we have transceivers, and those transceivers then send out a signal. That signal gets modified by the tracker that or the little tag that we have glued to the back of the beetle. And we listen back for that signal that comes back to us. So this is a technique that is one that we've developed. So we've been really pushing this forward. They have very flexible antennas on them. This is one of the first projects that we did with this technology. Pretty inexpensive. There's no battery on them, so they last really long. Unfortunately, there's no ID on it. But this allows us to track individuals in the environment and see what they're doing. So this is technology that was originally developed for backcountry rescue. So if you get stuck in an avalanche, they come and find you this way. The picture here is one of my technicians with one of the handheld units. I really want to have enough money to have one of the helicopters, but so far we're not doing that. We're doing the handheld units. This is searching in coffee, so actually not for the beetle. But one of the things that we were able to do with this, in talking to constituents, talking to farmers, one of the questions that we were getting was, where are the beetles hiding? They seem to be really cryptic. And that was one of the things that we were able to show is like, a lot of the time we would find them in these dead leaves. They'd be curled up in there and they would be hiding inside of them. And so some other pictures here of them on the trees. What we were able to do then too is kind of two different experiments. So the aerial picture that we see here is from out by Kamehameha schools, and the Kukui that they have at the front there. So we did studies that were over a number of days. So looking at kind of where they went. So this is a 10-day track here. This is a 7-day track here. Again, kind of looking at where do they spend their time. So unsurprisingly, they really like to be in the tree. We were also out there for some periods at night where we would just follow them as they would move around during the course of the night. And again, kind of struck by how much they walk, how little they fly. And even in getting up into the trees, how much of that is them crawling and not flying. The last thing that I'll talk about then is the citizen science mapping that we've been doing. So very dependent here on the community to be reporting. We did have a site. Unfortunately, ArcGIS has cut us off. And so we don't have a site at the moment where people can report beetle sightings to us. But let me explain a little bit about what we have here. So this is community members that are reporting where they're finding the beetles. So in 2020, which is when we started this, all of the red spots then are ones from that year. When we move on to 21, it's the red spots indicate again that year. Black ones are from the previous year. So as you move on, what I want to draw your attention to are the red spots here, and we can see that they're moving up the Hamakua coast. They're moving further down here into Puna. As we go along, you can see again further up the Hamakua coast, right through to 25. So there's some things that we got from that. One, if you look at the time of year that we were getting reports, those are largely in this kind of May to July area. Elevation, the vast majority of these are at low elevation. Now there are some caveats on that because we're getting information from the public, and it might be that there just aren't people living in the areas where the beetles are moving into. But for the most part, it looks like there might be an elevation part to where they're able to survive. So whether that's temperature as well or other factors that are going on to it, but just noting that elevation seems to be a factor. The other thing is this last graphic, which is maybe a little harder to understand, is about, when in each year, what percentage of finds are we finding in the existing range and how much of them are expansion. And generally what we're seeing is about a 25% expansion per year as we're looking at these beetles moving around. So just in conclusion then, the 4 different experiments that I've talked about, and again, we can take questions when others are done. But the idea here was, we really knew nothing about this beetle as it came in. And so we were trying to do as many different kinds of experiments to find out about their biology. So the phylogenetic analysis, we really hope that that gives us diagnostic tools that we can use to figure out, do we have Queensland longhorn beetles, or is it another cerambycid? The nighttime activity that we were looking at, we found it mostly sedentary, definitely night active. The field tracking was something that we used, again, confirming that they're super cryptic and where they're hanging out. They love to be in the trees, but also picking up that, a lot of what they are doing there is crawling as opposed to flying. The one piece that we're kind of missing from that flight tracking is the long-term movement. And we've been working with some drone development, which will probably help us to get to that longer term or those longer range movement in the future. And then finally, with citizen science, we know that their high populations are May and July. Generally, they're below 500 meters. And at this point, they appear to be expanding along the coast. So with that, thanks to the team who worked on this. So a number of different agencies and a whole bunch of people that were working on it. And if you do have questions, hang on to those. And after we get through the other folks, I would be happy to take some questions. So I will sign off. Cool. And Jade, back to you.
Yeah, thank you, Matt. That is fascinating. I didn't realize they were so, I guess, lazy. They don't even like to fly. That I didn't know that. That's kind of interesting.
They like to bite us, right? But they
Exactly. Yeah.
but actually, yeah, the amount of time that they spend flying so far from what we've been looking at, it's not that much. Of course, they don't need to fly that long to move out of an area. So we're very
That's
interested to look at
true.
that migration stuff eventually. So yeah. All right, I will quit.
Thank you, Matt. So next person, we're gonna get right into it. It's Dr. Roxana Myers, research plant pathologist specializing in, okay, I'm gonna screw that, nematology, study of nematodes, at the USDA ARS Daniel K. Inouye, again, a PBARC, where her responsibilities focus on the development of cost-effective and sustainable approaches to the control of plant parasitic nematodes. In addition to that, she also uses the entomopathic nematodes as a biological control agent against invasive pests, such as sweet potato weevil, 2-9 spittlebug, and Queensland longhorn beetle. Yeah, so all you, Roxanne.
Thank you, Jade. Really appreciate the opportunity to be here today to share some of our research with you. So I'll be talking to you today about entomopathogenic nematodes. And I added Kathy Mello as a co-presenter because she's really instrumental in finding these local isolates and also developing the injection methodology that I'm going to be sharing with you today. Oh, okay. Sorry about that. So I just want to start out just by sharing what a nematode is. They're one of the most abundant and diverse multicellular animals on Earth. There's over 30,000 described species, and they fill every ecological niche on our planet. But there are very different types. Some are just plant parasites that feed on the roots of crop plants. The ones we'll be talking about today are insect parasites. There's animal and human parasites, as well as free living. So insect parasites are microscopic non-segmented roundworms. They attack insect pests only. They have a very broad host range. They're easy to apply. They're safe for humans, animals, and plants. There's no personal protective equipment required, and they're not regulated by the EPA. So how do these nematodes control insect pests? Infective juveniles will seek out an insect host, usually a larvae. They have a symbiotic bacteria that is released. The insect dies and the nematodes begin to develop. and reproduce in the host, and then the next generations of infective juveniles emerge. We followed in the footsteps of Dr. Arnold Hara and Harry Kaya, who originally did a survey for entomopathogenic nematodes in the state. We went to the exact same places, and we were able to uncover the species Heterorhabditis indica on all the main islands. So here's Kathy and her husband, Joey, which some of you may know. He is retired from DLNR. So these nematodes like the coastal areas, so that's where we found them in very sandy soils. So this species is highly virulent against a broad host range. It's an active hunter, which means it cruises through the soil following chemical cues from its prey. We've tested it on over 40 invasive insect pests, including small hive beetle, banana moth, coffee berry borer, and we're currently working on a project with two-line spittlebug. One of the successes that we've had is using these nematodes to control the sweet potato weevil. We recently completed a NIFA ORI grant with the University of Hawaii and Auburn University to run field trials with these EPNs. And the nematodes will actually go inside of the tubers and hunt down the larvae. And then the next generation will come out and move on to the next tubers. And in the simulated field trials, we had a 77% reduction in the sweet potato weevil populations. So we thought, how can we use these nematodes to try to control Queensland longhorn beetle? So that's when Kathy came up with the idea to inject the nematodes directly into the galleries that the beetle larvae are creating inside the trees. And how we do this is by using a disposable plastic syringe with a 1.5-inch needle. We have the nematode solution. We pour it into a container and make sure you stir it very well and agitate the solution before you draw it up into the syringe or transfer containers, because the nematodes do settle. We found that it's best to apply the nematodes early in the morning, at dusk, or on rainy days. And always store your nematodes in a dark, cool location until application, but use as soon as possible and do not refrigerate. So the first step into doing this application would be to scout your orchard for QLB damage. You'll look for sawdust-like frass, oozing sap, girdling of the trunk, and lifting of the bark. If you see the adult emergence holes and nothing else, then that means that the adult beetle has emerged. And if you don't see any other signs, active signs of infestation, then it's too late to treat at that point. So this is a video that we made where we wanted to kind of show what the frass looks like. You see what some of this sawdust looks like, and also this spongy bark. That means that it's hollow underneath. The larvae has already fed on the inside. So we would go out, we would scout it, we would tag it. So when we have the nematodes, we're ready. We know exactly where to go to inject the nematodes. And so this is the injection process. You will search around to find a soft area where the needle will go directly in, and then you'll slowly push the material in there. And then once you start seeing it coming back up or draining out the other side, then you'll stop. But we try to put as much of the solution as we can and until we fill the gallery. And that gives the nematodes the best chance of being able to hunt the larvae down. So one of our first field trials we did was at the retired extension agent from CTAR. We went out to her field. She had a really bad infestation on kukui and citrus, and we were very excited after 4 months to go back. After one treatment, we didn't see any signs of the QLB activity, and we did apply it on the most rainiest day of the year, and must have been very good for the nematodes. We were very excited to see that the new growth was coming back. So after that, we started going out to a number of different farmers and teaching them this technique. We've done quite a bit of outreach and online trainings. We've worked with a lot of grower groups, including the tropical fruit growers, cacao associations, and the Hawaii ʻUlu cooperative. So then we partnered with the Big Island Invasive Species Committee to assist us with the EPN rearing and distribution. And so they're currently providing the EPNs at no cost to growers and community members. One of our bottlenecks is ramping up production of the EPNs, but we are working on that on our end as looking for techniques for mass rearing and optimizing the host insect rearing as well. So if you're interested in getting on the list to get some of these nematodes and do an application, please contact Jade Miyashiro, and she will get those out to you. So we're really grateful for BISC for helping us get these nematodes out to the farmers. I'd just like to thank my nematode team and Tracy Matsumoto, who was very instrumental in getting us to work on this project. And she always believed that this would be an effective method. And her daughter, Kaylee Brower, helped us to make the video. So, and thanks to Stacy Chun, who's done a lot of the outreach events for us. We're very grateful. Let me stop share.
Thank you, Roxana. It's awesome. Those things are so cool. I was actually spending this morning cleaning mealworm cages, which I need to finish right after this.
Well, thanks for doing that. We really appreciate it.
Of course, no problem.
And to Emma for taking the next steps, which she's talking about.
Yes, exactly. So yeah, so next person coming, we have Emma Stierhoff. She is the lead ecological research technician for Liko na Pilina. She graduated from Hilo with her MS in Tropical Conservation Biology and Environmental Science 2024. Since then, she's been leading data collection and maintenance of a Kōkaha-based long-term restoration site with the Liko na Pilina hybrid restoration project. As QLB became a pressing problem in the Kōkaha forest, Emma and her team started working with folks from the USDA to implement the nematode control to save important canopy trees in that forest. And I believe they also brought to our attention how QLB has started going after some native plants that we weren't aware of. Go ahead, Emma.
Thanks so much, Jade. Aloha kako. Thank you so much for tuning in today. So I will be talking about the effectiveness of this nematode biocontrol that Roxana laid out really nicely for you against the Queensland Longhorn beetle, based on an experiment we carried out at our Keaukaha site, and then some next steps for this research. So just to give you some background on Liko na Pilina and how we got into this work, Liko na Pilina is a hybrid ecosystem restoration project through the University of Hawaiʻi Hilo. And hybrid ecosystem basically means that along with native plant restoration, we also use non-invasive introduced plants that can complement those native plants. This is a really heavily invaded area, and so we really needed to use faster growing non-invasive introduced plants to create shade and slow down nutrient cycling to reduce that encroachment from invasive plants. And this project started with outplanting in 2013 at the Keaukaha site. And since then, we have seen reduced encroachment from invasive species. We've also seen greater recruitment of our desired plants, like our outplants, as well as some existing native plants like ohia, llama, and kolea. And this approach can also be used to address variable management needs in Hawaii and the Pacific. Often going back to that historical state is not possible with the amount of invasion that we're dealing with. But these non-invasive introduced plants can be used in tandem with our native plants to still promote native diversity. So we got into QLB because we noticed many of our kukui, especially dying as a result of Queensland Longhorn beetle. Most of our mature kukui trees at the site are now dead as a result of QLB infestation. And so we really needed to address this. These are important canopy trees for our site. They really help create that nice level of shade to keep the invasive plants out and allow the native plants to reproduce. And so in a need to address this, we started working with USDA to implement the nematode biocontrol in 2024. And we also saw, like Jade mentioned, some possible infection of native species. We've seen what looks like exit holes in Alahe'e and Hawaiʻi Ohe. but these infections haven't been confirmed. It's just a speculation at this point. So in the summer of 2025, last year, we worked with a PIPES intern, Lila Shapiro, to do a experiment testing the effectiveness of the injection treatment that Roxana so beautifully laid out for us on Kukui and ʻUlu trees at our site. And so we did a health assessment and an infection assessment of all of our trees. So we rated infection level from 0 to 3, with 0 being no active frass, 1 being kind of a moderate or mild infection, 2 being that moderate infection, and then 3 being a really severe infection where over 30% of the tree is exhibiting frass and other symptoms as a result of QLB. And so we assessed the trees, gave them the treatment, and then reassessed them after the treatment. And we found that with Kukui, we tested 62 trees that were infected with QLB. And the majority of these trees were either able to completely eliminate QLB or at least reduce their level of infection. And then at the worst, just a small percentage of trees, about a quarter, had no change in infection levels. So they never got worse at the very least. But almost half of the trees were able to completely eliminate infection. These results did vary based on the initial infection level of the tree. So trees that had a lower infection level were more likely to get to that infection level of 0 than, say, trees with an initial infection of 3. It was more variable. A lot of them stayed the same, whereas only about a third got to a completely eliminated infection. And then looking at ʻUlu, we had fewer ʻUlu that were infected at our site. They definitely seem to prefer the Kukui. But we tested 11 infected ʻUlu at the site, and the vast majority of these had no sign of infection after treatment. And just a few had a reduction or no change in infection level. So again, no trees got worse after nematode treatment. And again, the results varied slightly based on that initial infection level. So if the initial infection level was 1, 83% of those trees completely eliminated infection, whereas an initial infection level of 2, only about 60% completely eliminated, and the remaining stayed the same or at least improved. So ultimately, we found that this injection with entomopathogenic nematodes does help reduce the QLB infection in Kukui and ʻUlu trees, which is really promising. And this effectiveness does depend on the initial infection of the tree. So this really highlights that need for surveying and early detection. The earlier you can catch QLB in your tree, the more effective this treatment is going to be. And many of these trees did require multiple injection treatments to eliminate QLB. So knowing now that this injection works and it works pretty well, there are still some limitations to implementing this. One being nematode availability, just increasing nematode production. Bisq is doing a lot of work on this and making them available to community members, which is really amazing, but it takes time and a lot of time really just to rear them. It's not a super high cost thing, but it takes time to rear them in captivity. And so Roxana and her crew are working on improving rearing methods and looking at mass rearing methods to make them more available. And then another big thing is the time intensiveness of this treatment, especially if you do need to apply it more than once to a tree. This injection method does take a good amount of time. You need to, find the pukas in the tree and inject it into there. So that can be a limiting factor when you have, hundreds of trees in a cacao orchard. It's not necessarily doable. And then we're also working on understanding the best conditions and methods for treatment. Roxana mentioned it's best to do on rainy days or at dawn or dusk. But if we can figure out, is it better to do it at dusk when the weather's getting cooler? Or is it better to do it at dawn as the day is starting? Really figure out when we can apply these to maximize the effectiveness. And also understanding the QLB life cycle, like Matt mentioned, we really don't know much about this beetle, and he and his crew have found out a lot about the adult life cycle, which is really awesome. But we still don't know a lot about what they're doing in the tree, how long they're an egg, how long they're a larva, how long they pupate. So it would be really helpful to understand all of these things to know what time of year we need to look for these signs and when we should be treating. So the future research that we are going to focus on is determining other possible methods of nematode application that might be more effective and or faster. So we'll be testing this injection method alongside a method with a nematode-soaked sponge that kind of gives the nematodes more moisture going into the tree so that they might survive longer on their way to find a host. And we'll also try an inoculated cadaver, which is that larva that has the nematodes breeding inside of it before they emerge. And so we can put that in the tree and see if, having these really fresh nematodes emerging from the cadaver going into the tree can be effective. And these methods have been tested with other nematode biocontrols and found to be highly effective. So we think it's worth seeing if they work here. So the scope of this research is kind of over the next year. We're looking to carry it out. So we'll be testing treatments on these different treatment methods on kukui, ʻUlu, cacao, avocado, lilikoi, and citrus, and maybe durian as well. We'll be working in partnership with the folks at USDA at the germplasm by Panaeva. And as we carry this out, we're hoping by January of 2027, we can, compile our findings and report those out to determine the best practice for this application. And then in the remaining months of the next year, from February to May of 2027, offer workshops on these updated nematode application methods and support growers in treating their trees. To make this as accessible as possible, we want people using these nematodes that BISC is providing. And you can all help with this. This project is possible because community members like you reached out to legislators and said, this is an issue for me. This is something I care about that needs to be addressed. And you can continue to do that, continue reaching out to your legislators and encourage them to allocate resources towards research and management, because that's what allows us to continue doing this work and understand this pest and how to mitigate it. So mahalo nui to everyone who has supported this work. We have a whole nematode hui of people from USDA, BISC, US Forest Service, and Hilo working on this. It really takes a village to make this happen. So mahalo to everyone who's collaborating on this. And mahalo to our nematode partners. Here they are wiggling under the microscope. And feel free to reach out if you have any questions or are interested in learning more.
Yeah, thank you, Emma. That was awesome.